Alright, hello, welcome to Teaching Bio. Today we're going to look at the entirety of photosynthesis in under 15 minutes. So we're going to start off with looking at the structure and function of the chloroplast. So this lipid droplets, the chloroplasts have these to sort of store cholesterol and use them for other biochemical processes. It's got the stroma. Now the stroma is basically sort of like this matrix, and that's where the light-independent reaction will happen, and this contains enzyme that will fix carbon dioxide to form glucose. This is the thylakoid, they arrange in stacks, and that's where the light-dependent reaction happens, and this is where the chlorophyll pigments that absorb the photons of light remain. This is the inner membrane. This is the granum, which are effectively stacked thylakoids. So a stack of thylakoids make a granum. So the advantage of having a granum is that it's more efficient. Here we've got starch grains, which will store excess glucose effectively made during photosynthesis, and then you can use that as a source of respiration. This is the outer membrane, with the phospholipid bilayer to control the movement of gases and other molecules in and out. And then finally, we've got ribosomes. So we've got membrane bound and free, and the ribosomes will obviously produce the relevant enzymes needed, such as Rubisco for the light independent reaction, as well as other pumps needed in the light dependent reaction. So we're going to look at the light dependent reaction first, which is split into these steps. First step is photolysis. So here's a chlorophyll molecule. What will happen is the chlorophyll will absorb photons of light, and the chlorophyll molecule contains electrons. When it absorbs photons of light, it oxidizes the chlorophyll, and that excites the electrons in there. So because they're excited, they basically get released to a high energy level, and then the energy that is released from here will be used to split water into 2 protons and half of oxygen. The oxygen will diffuse out via the stomata, and then the excited electrons get taken up by electron carriers, which are basically just these molecules. This process is called photoionization. The electrons that are released from the lysis of water will be attracted to the positive charge of chlorophyll, and so it will bind to it. So these electrons will then replace the electrons that have been lost. So again, here's the full picture. Photons of light absorbed, excited to a high energy level, use that to split water, oxygen out, protons to the proton pump, electrons from here move back in, replace the difference. Then we have the electron transport chain, or transfer chain. The excited electrons that were taken up by the electron carrier are then passed on to many more electron carriers, and that's the electron transfer chain. The electron carriers accept electrons and are reduced before losing electrons to be oxidized, and then they pass along. So there's a series of oxidation reduction reactions. So remember, oxidation is loss and reduction is gains. When this electron carrier will gain the electrons, it will be reduced, and then when it loses it, it will be oxidized. And then as it moves along, that will lose energy, and that energy is very important, which we'll look at in a bit. So again, this is the first step, photolysis. Energy level taken up, it'll get reduced, pass it along, it'll be oxidized, this will now be reduced, and it'll carry on and pass it along, and it keeps going along. So we're going to have a look at the proton pump now, and this is where the energy that we kept losing from the electron transport chain will be used for. So the energy lost from the transfer of electrons will be used to pump protons into the thylakoid space. So now the energy that we lose from the electrons we've been moving along, we'll get to move protons into the space. Remember the 2 protons from the start of photolysis, they also enter the thylakoid space. That will then create a higher concentration gradient of protons here compared to here. Now we're going to photophosphorylation. So the protons that we had in the thylakoid space, they're now going to move out because we created a gradient. Remember, high, low, down the gradient, it will move out. So it will move out of the thylakoid space. And when the protons move out, they combine with ATP synthase, altering its structure. By doing that, we make ATP synthase effectively ready to produce ATP by allowing for ADP and inorganic phosphate to combine, and that's called photophosphorylation. Phosphorylation because we're making phosphate, well, ATP, and photo because we're using the photons of light, and this is called photosynthesis, and that is a condensation reaction. So again, just to recap, electrons, it'll be reduced, then move it along, oxidized, reduced, carries along, loses energy. Energy is used to pump protons into the thylakoid space, creates a gradient, protons move back out, moving through ATP synthase to produce ATP. And we also have this component here, NADPH, or otherwise known as reduced NADP. So at the end, another chlorophyll gets hit. You don't know the names of the chlorophyll, that's too much detail. The electrons get excited, they move along, and the same process effectively happens. Then it gets taken up by electron carrier and it gets reduced, and it passes down a bit of a shorter electron transfer chain. And then eventually that will go to NADP, and then NADP is attracted to electrons because there's a negative charge, like attracts like. It will then sorry, the negative charge will repel each other, but the electrons will be taken up to NADP to make a negative charge, and then the protons will be attracted to the negative charge. And then because of that, we'll get NADPH, otherwise known as reduced NADP. Okay, NADPH. So this is the whole thing in detail. So again, here the protons moving down. They move down because it's the gradient. The gradient's made by the loss of energy from the electrons, and they pumped in with the proton pump here. Now, what's the point of doing all of this? Well, the point is that we transfer light to chemical energy, ATP, and we need ATP. And we also produce NADPH. And NADPH is very important for the light-independent reaction. So this is known as the Calvin cycle, and this is effectively the production of glucose. So this is the Calvin cycle. You don't have to know the names of all of the different immediates. You've got to know some things like glycerate 3-phosphate and ribulose bisphosphate and triose phosphate. But you don't have to know any sort of other names that are mentioned. Like these are just the sort of, you don't have to know, there's a lot more detail to this. You don't have to know all the different names there, but it probably helps to sort of understand it in terms of the bigger picture. So we're going to look at the first stage of the Calvin cycle. So carbon dioxide, which will move in to the plant from the stomata, will bind and react with this compound called ribulose bisphosphate, which has 5 carbons in it. And then we need to use the enzyme Rubisco to catalyze that. So once that gains carbon dioxide, it will form a temporary 6-carbon molecule, which is unstable, and that breaks down to form 2 lots of glycerate 3-phosphate, which have 3 carbons each. 2 times 3 is 6, so therefore that totals the 6 we made. Now, glycerate 3-phosphate is going to go through a couple of reactions. This is where we use the NADPH that we produced. So NADPH is now going to send its electrons to glycerate 3-phosphate. So this also means that NADPH is being oxidized and glycerate 3-phosphate is being reduced. Once it's been reduced to accepted electrons, it will go on to produce triose phosphate. This actually needs an input of ATP, which is why the ATP that we made in the transfer chain is used. So we've got to put some energy in, and again use NADPH to make 2 lots of triose 3-phosphate. Triose 3-phosphate is a 3-carbon molecule, hence the name, triose, 3. 2 times 3 is 6. The maths make sense. We get 2 lots of 3-carbon molecule. So the 2 molecules of phosphate make 6 carbons in total. Only 1/6 of the carbons from here, so 2 times 3 is 6, and 1 of them will be used to make glucose. And the remaining 5 will be used to regenerate ribulose bisphosphate. Remember, glucose is a 6-carbon molecule, therefore we will need to turn the cycle 6 times to get a full molecule of glucose. So we're going to regenerate ribulose bisphosphate, and again that will require an input of ATP as well. And then we can finally reform ribulose bisphosphate, which can, guess what, combine with carbon dioxide again to form 2 lots of glycerate 2-phosphate. So this is sort of the whole picture in detail. So again, the chlorophyll absorbs photons of light, excites, electron transport chain, lose energy, use that to make ATP, and NADPH. They're fed in. It's a light-independent reaction, so we can make glucose. And the products of this, so the ADP and the NADP, can go back and be recycled. So it's this brilliant sort of cycle that works together nicely. So we need 6 turns of the cycle to make 1 molecule of glucose. We need 6 molecules of NADPH and 12 molecules of ATP. It's important that you remember these because they will definitely ask this in some form of way, maybe in a graph. They don't use maths with it, but it's good to know these numbers. 2 required practicals in photosynthesis topic, and they are chromatography and the factors affecting dehydrogenase activity, and I've made videos on them that you can find on my channel. Okay. Another thing you need to do is you need to be able to identify environmental factors in related to agricultural practices. So there's 3 sort of factors that limit the photosynthesis. This is very basic. It's basically just GCSE. First one is light. Obviously, you need light. The more light, the more photosynthesis, and that's needed for the light-dependent reactions. Getting photons of light to excite the chlorophyll. There's the graph you need to be able to remember. High CO2 obviously means a faster rate. Low CO2 means slower rate. Carbon dioxide obviously needed because we need to fixate the carbon dioxide to ribulose bisphosphate to make our 2 lots of glycerate 2-phosphate to then go on to make glucose. And these are the 3 graphs. Again, the ideal conditions will be having decent light, good temperature, and high carbon dioxide concentration. Finally, temperature. The graph will look like this. Again, it's basically similar to the normal enzyme graph. The more heat we have, the more heat energy means that there's an optimum rate for the enzymes. We get more enzyme substrate complexes. So the more complexes we have, the faster we can basically produce glucose. But obviously it gets to a point of the optimum, then the enzyme will start to denature. Again, at too low temperatures, there's not enough heat energy to allow for successful enzyme substrate complexes. So at any given time, there can only be one factor that actually limits it, and that's known as a limiting factor. You can't have more than one limiting the rate of photosynthesis. It'll just be that one factor at that moment in time, because it's not enough, or it's too far away from the optimum that we need. So, we're not enough. Now, you might be asked to evaluate data. This is very likely, and these are all very boring exam questions, but really they're very simple. It's just, if you use logic, you can work them out. And it's basically just, if you can remember this paragraph here, you'll be able to answer any question involving data for agriculture. So just note that in a closed greenhouse, the CO2 concentration can fall low, so we can artificially increase the CO2 concentration. And that will increase our photosynthesis. How do we increase CO2 concentration? Well, obviously, it's just going to be combustion. So this is just, foundation GCSE chemistry. We just burn a fuel, we release CO2. That in turn will raise the temperature as well, because we're releasing heat energy. And then how can we increase light? Just turn the lighting up. So upon that you understand this graph as well, this kind of can look a bit complicated, but really it's quite simple. It's just rate of respiration, net CO2, and this is how it increases. There's the maximum rate of net photosynthesis at that point there. We have the light saturation point there. This is the light compensation point, also known as the equilibrium point, where the net CO2 uptake will effectively be zero. So the amount of CO2 that we output from respiration will equal the amount of CO2 that we sort of take up for photosynthesis. Thank you for watching, and please subscribe. Any comments, please let me know if you have any questions, and just leave them in the comments. Thank you.